Hi everyone, and welcome to our video on the Hardy-Weinberg Principle. What we're going to be doing in this video is carrying on with our Module 6 Part 2 series on patterns of inheritance, and we're going to be delving into another one of these calculation type questions that can come up. So what we're going to be doing in this video is having a specification reference 6.1.2f, the use of Hardy-Weinberg Principle to calculate allele frequencies in populations. Do note, you don't have to memorize the Hardy-Weinberg Principle equations, you just need to obviously know how to use them. So you'll get them provided for you on the exam should you need to use them. Okay, what we're really looking at this point is something we term population genetics. And that's where we're actually going to study how the different alleles frequencies will change in a population over time. So what we're really wanting to do here is basically track and see how the frequency of alleles changes over a period of time, maybe down to changes in the environment, etc. So it gives us a way to have a look at this. And there are several factors that we need to bear in mind here. So population size, migration, the rate of mutations, any environmental changes that are happening, the founder effect, etc. The list goes on, you can see them all here. These are all factors that are going to influence, obviously, the alleles that are present within a population at any given time. Now, it is worth dropping back into some of our work that we did back in Module 4, where we had a look at the definition of a species, and hopefully we do remember that if we have got two organisms that cannot interbreed and produce fertile offspring, then we are going to have to define them as two individual species, because if we've got these two species that have now become so genetically different from one another that they can't interbreed for a reason, then they've undergone that process of speciation, and as a result we now have two different species. And it is worth reminding you that when we're talking about the definition of a species, remember, specificity is key in your language here. We are talking about two individuals that are able to interbreed and produce fertile offspring. Don't forget the second part to the definition of the word species there. Okay, let's get to the nitty-gritty of the Hardy-Weinberg Principle then. What we're looking at here then is a way that we can actually determine the different allele frequencies. So if we wanted to know the proportion of a population that are carriers of a particular recessive allele for a disease, for example, this is what we would be using, this Hardy-Weinberg Principle, in order to work it out. Before we delve into the mathsy bit, let's just recap on some of these key assumptions that are made when we're using the Hardy-Weinberg Principle. First one is the population is large. Secondly, we have random mating occurring. Third, there's no mutation, migration, or genetic drift occurring. And finally, that there is no selective advantage for any particular genotype. So one of the common questions that comes up on the exam papers does have a look at basically these assumptions. Now, it might just explicitly ask you what assumptions are made, or it might ask you why the Hardy-Weinberg Principle may not be very good to use in a given context. So obviously, if we're looking at why Hardy-Weinberg may not be appropriate to use, then look and see in the information they've given you at the start of the question, how big is the population we're talking about? Are they able to randomly mate? So if we're talking about obviously a zoo population, then it's not really going to be a large population. We're not going to have random mating occurring, and therefore Hardy-Weinberg really shouldn't be applied in that context. Okay, so when we think about the Hardy-Weinberg Principle, one thing you've got to remember is what does P stand for and what does Q stand for. They will give you the equations. They will not give you what P and Q stand for. So make sure you remember which is which. So P is the frequency of the dominant allele, and Q is going to be the frequency of the recessive allele. Now, what we do know is that at any particular gene locus, then we have two alleles and two alleles only. Therefore, P plus Q must equal 1, because obviously if we've only got the two potential combinations, then add them together, that's got to come to 1. if we just apply that first part to an example question then, if we've got a population of 100 individuals, then we are going to have 200 potential alleles present. Now, if out of those 200 alleles that are present, 140 of them are dominant alleles, the frequency of dominant alleles in that population is going to be 140 divided by 200. So obviously the dominant divided by the total number gives us 0.7. So if we then wanted to work out our proportion of the recessive alleles, we know that obviously P plus Q equals 1. Therefore, we can do 1 minus our p-value of 0.7 gives us 0.3, the recessive. So we can obviously use the information in the questionnaire to work out either the frequency of dominant alleles or the frequency of the recessive alleles. So we can work out both from that initial information. Now, there is a second equation that we need to be aware of for Hardy-Weinberg, and this is going to allow us to work out any probability of a particular genotype in the offspring. And what we have at the bottom is this second equation. So p squared plus 2pq plus q squared equals 1. Now, what we need to do then is break this down to understand what we're talking about. So first of all, the p squared is relating to obviously dominant alleles, because we know that p is dominant. And if it's p squared, that means that both of the alleles are going to be dominant. So what we're looking at is the homozygous dominant offspring. Now, the reason it's p squared is because if we've got, obviously allele for dominant is p, and we've got 2 of them, then it's going to be p times p, which is just p squared. The next bit that we're going to look at is the other end of the equation, the q squared. Now, q, remember, is the recessive allele. And because it's q squared, that tells us we have got homozygous recessive, because q times q gives us q squared. And the last one there, 2pq, this is our heterozygous combination, because when we're looking at this, the reason it's going to be 2pq is obviously we can have a dominant and a recessive, but it could go either way round, remember. So you could have obviously one dominant and one recessive, or it could be the other way. So 2 different possible combinations of those heterozygous offspring. So as a result, p times q gives us 1, and then there's going to be 2 potential combinations there, and therefore we times it by 2pq. Okay, I think one of the easiest ways to get to grips with Hardy-Weinberg is to have a look at a few example questions. So I've got a couple for you here looking at 2 different aspects of using Hardy-Weinberg to actually carry out the question they could ask you on the exam. So here's our first one, okay? What we've got, first of all, because this is an exam question, they are going to give us the equations for Hardy-Weinberg. Then we get our information about the context of the question. So make sure you read this carefully, and I suggest highlighting or making notes to yourself about the key bits as you go. So if we have a little look, albino rabbits have white fur as these individuals are unable to produce the pigment melanin. The ability to produce melanin is controlled by a gene with a dominant allele, capital B, resulting in brown fur. So brown fur is going to be our dominant trait. The recessive allele is albino, okay? So what we're going to see here is any of our albino rabbits must have obviously both lowercase b's. Okay, so they've got to be homozygous recessive to be albino. If we're talking about brown fur rabbits, they could obviously be either homozygous dominant or they could be heterozygous. Okay, so what we need to think about then is the different genotypes that exist. The next bit of our question there tells us of the 60 rabbits that we have in a pet shop, 45 are brown. So a student decided to use Hardy-Weinberg to estimate frequencies of the alleles in this group of rabbits. We need to use Hardy-Weinberg to calculate the frequency of the dominant allele in the group. Okay, so that is what we are going to be doing. Now, just going to make it a little bit smaller so I've got some space to do some writing at the bottom here. Okay, remember what we said. Okay, so albino is our recessive. So in order to be albino, they are going to have to be homozygous recessive. To have brown fur though, what they could have is either homozygous dominant or heterozygous. So what we're told is our total population is 60 and 45 are brown. So what we can do then is, you might think, great, if I want to know how many dominant alleles there are, we'll just do 45 divided by 60. You can't do that, because we are just looking for the frequency of the dominant allele, and that on its own is not going to give us it, because remember, we've got two possible genotypes there. So we're not actually going to be looking at the bit they've explicitly told us. We need to think about how many of these are albino rabbits, because the only way you can be albino is to be homozygous recessive. So if 45 out of 60 rabbits are brown, then 60 minus 45, hopefully we can do that maths in our head, is 15. So we have 15 albino rabbits. Now what we need to do is work out, obviously, our Q squared. So in order to do Q squared, which is our homozygous recessive, we are going to do 15 divided by total population of rabbits, which is 60. And if you plug that into your calculator, that should come out at 0.25. Now, that is Q squared, remember. And what we actually want is Q. So to get Q, we need to take the square root of 0.25. And if we do that on our calculator, you will end up with 0.5. Okay. What we need to work out though is the dominant allele, remember. So we're going to come up to this second equation there, P plus Q equals 1. So if P plus Q equals 1, we've obviously got to work out P, but we do know that Q is 0.5. So all we're going to do there, rearrange your equation, 1 minus 0.5 equals our P value. So 1 minus 0.5 is 0.5. So that gives us our final answer. Now, whenever you're doing this, it's always worth setting out the working nice and clearly so that if you happen to make a silly calculation error, we can amend it and obviously see where you've gone wrong. Okay, second example of a question then. We've got another one from past exam question here looking at Bengal tigers. So we've got 1 in 10,000 are going to be white, and it tells us that the allele for white fur is recessive. So what we've got, Hardy-Weinberg equations again, we then need to work out the percentage, okay, and that word percentage is going to be important, of our Bengal tigers that are heterozygous. And we're going to give our answer to one significant figure. Okay, again, let me make it a bit smaller so I've got space to actually do the working. So first thing, we know obviously that we're looking at our recessive trait first of all, and we're told that 1 in 10,000 is white. So we can do our 1 divided by 10,000 to work out obviously our Q squared. Okay, so that's going to give us Q squared. Remember, we're always going to aim for Q, so what we're going to do is the square root of 1 divided by our 10,000, and once we've actually worked that out, we're going to end up with 0.01. Okay, so Q is 0.01. We can then work out P by using this. Okay, so again, we've got 1 minus 0.01 equals 0.99, and that's our P value, okay, our dominant. Okay, that's not the end of this one though, okay, because what we're looking for is the heterozygous. So heterozygous, remember, is this bit here, okay, so we're using the other equation at that point. So in order to work this out, we've got to do 2 times P times Q. So 2 times our P value, which is 0.99, times the Q value, 0.01. Again, plug it into your calculator and you're going to get 0.0198. Okay. Go careful though, just thinking that's the end, because remember we had that word percentage here. So this isn't a percentage. We then need to change that to a percentage. So 0.0198 times by 100 is going to give us 1.98%. 2%, but we're still not done because it tells us to give our answer to one significant figure. So that should be 2%. This is where reading the question really comes in, and that highlighting and double checking that you've done everything it's asked you to do in the calculation is going to be critical. So do make sure that what you're doing is double checking when you think you've got to the end, that you have actually got to the end and you've done all the bits of the question that is asked you to do. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the website where you can find a range of other resources to help you in your A-level biology studies.